Thank you, Town Frederick, for the invitation. It's a pleasure being here and learning what you're doing. Quite impressive what you're doing in the biotechnology area. Let me give you a little bit of a background of how we came to work with this organism that degrades these perfluorinated alkyl substances. See this one? No. Here's one.
Click again, and then you can.
Okay. Okay. Good. Of course, the work is done by my students and postdocs, not by myself. So I need to acknowledge their work. This goes back almost 20 years. Field work close to Princeton in an area that we call the Pine Barrens, which is rather acidic, iron-rich soils. And here you see some example where a tree topped over. It's a riparian wetland. You see how where the roots come down and pump oxygen into the soil, how the iron gets oxidized. On the left here is a picture where you have iron water, groundwater with iron coming to the surface as the water touches oxygen, the iron oxidizes. And doing field work, that's a long story, but we noticed that ammonium was being oxidized under iron-reducing conditions. So we postulated that this was a feasible reaction in a 2005 paper. A little bit later, this fellow in Japan, Soyama, found something similar in a biological reactor, and he called the term FIAMOX. Since then, the multiple papers that have come out where people have noticed this sort of ammonium oxidation under iron-reducing conditions. We went back a couple of years later to that same site, and at that point, knowing that my biology wasn't this strong, I hired a microbiologist, and we identified the organism that was responsible for this sort of ammonium oxidation under iron-reducing condition. It was an Acidobacterium. It is a novel bacterium, an anaerobic, it's an autotrophic organisms, anaerobic autotroph, they're slow growing, doubling time 10 days to 2 weeks. It's not a domesticated organism. We grow it, it eventually dies on us, and we have to go back to the field site and re-isolate it. It needs to have solid iron phase. It doesn't, if we add like Geobacter or so, you can add a ferric iron or ferric citrate or ferric chloride. It will use that as the iron electron acceptor. This one needs the mineral phase. The other thing that's interesting is if we grow it on hydrogen for a few generations, we cannot go back and grow it on ammonium. The enzyme, it's a oxygenase to oxidize the ammonium is on a plasmid. It loses the plasmid when you grow it on hydrogen for a few generations. Anyway, going back, we did then lots of analysis. We worked with the Chinese Ministry of Environment. They sent us a lot of samples. And what we found is that the organism is present in many locations where you have iron-rich acidic soils. Usually at very, very low numbers. Many times when we did DNA extraction, we didn't see it first, but once we added iron and we let the sample sit for 10 days, 2 weeks, and then do an extraction, we sort of start seeing that it's present. Eventually, we isolate the organism with the DNA sequencing, and we realized that it has genes for multiple reductive dehalogenases. I call it here a reductive dehalogenase homologue, RDHA. It has a fluoracetate dehalogenase homologue, and then it has 2 haloacid dehalogenase genes. It also has fluoride transporter genes. Fluoride is toxic to organisms, so even organisms that are in fluoride-rich groundwater have to have a fluoride pump to get rid of the fluoride, otherwise it's toxic. So this one has 2 fluoride pump genes that I'm going to discuss a little bit later. There is, once we did some searching, there is an organism, it's also an Acidobacterium that was isolated from the Black Sea sediments, TMED77, that has a very, very similar reductive dehalogenase as this RDHA. Otherwise, this RDHA seems to be fairly novel. Okay, so the question then is, can this acetylmicrobium defluorinate PFAS? And here we go to we did multiple incubations. I'm going to show you some cases when you see here on the left, these orange flasks is sort of how things look at the beginning. We sort of add ferrihydride. As the incubation progresses, it becomes more transparent gray as the iron 3 gets reduced to iron 2. And what we've done over and over is we have multiple of these vials, and we sort of sacrificially sample them to look at ammonium removal. When we add PFAS, what is the PFAS degradation? The headspace has to have a little bit of CO2. It's anaerobic. We pump it with a 80% nitrogen, 20% CO2 mix, because the organism needs that inorganic carbon to grow. This is sort of a little bit the summary of what we had in our sort of 2008 papers. It's slightly different results. What we see here is for all of these perfluoroalkyl acids, how they decrease over time in 100-day incubations. And we get something very similar for the sulfonates. And what I show here on the right is then as they decrease over time, we see the production of fluoride increasing, which is sort of, it's nice not to just report that something disappearing, but you want to see products appear. So fluoride, sometimes we can do a fluorine mass balance, sometimes not. When the pH gets a little bit higher, the fluoride and the iron 3 form a complex that is quite stable, so we cannot quite recover all the fluoride. Here, the next slide is for PFOA. As PFOA decreases, we see gradual appearance of a shorter PFAS, perfluoroalkyl acids. What is interesting is we see here this HPFOA being built up and then disappearing. It's the PFOA with one fluorine missing. And we worked with colleagues at the New Jersey Institute of Technology who had a nanosims LC-MS. So you after the compound comes out of the LC, you bombard it with high energy, you break the molecule into multiple fragments, and then look at the individual fragments and you try to put it back together. They showed that the fluorine that is missing is at two locations: the alpha carbon, the first of the functional group, and then the delta carbon, which is the third to the last. And those are actually the ones that have the lowest bond energy. Anyway, it's, again, you can get a reasonable fluorine balance if we look at the amount of fluoride produced and the amount of intermediates produced, based on the difference, the decrease in our PFOA. All right. So this is what I mentioned previously. We work at NGIT. So these are two points of defluorination. And then the next question that I'm going to address, just going to give you a flavor of what we're doing, is how do we deal with the large amount of iron that we have to supply? When you look at the stoichiometry, per mole of ammonium that I'm oxidizing, I need 6 moles of ferric iron. Our little flask, we add a lot of iron. When we had a continuous reactor, after a few months, it was just a paste of iron. It's very hard to separate your cells from the actual iron. So what is interesting is that well, so surface soils, it's very easy to mix iron. A little bit deeper, it's a little more challenging to sort of supply the iron. I'm going into that, and then in terms of bioreactors, is that my iron builds up because I cannot separate the biomass from the iron phase. So what is interesting with this Acinetobacterium is that we found out that it's electrogenic. So it is like Geobacter or Zionella, able to sort of transfer the electron to an anode as opposed to the iron phase. So we can grow it in microbial electrolysis cells. So we have an anode, a cathode. We put a little bit of a potential between the anode and cathode, and then we can transfer electron to the anode. We have grown it in these microbial electrolysis cells. So here it shows over time that it starts to produce current. This is the current density. The top is for the pure culture of the Acetobacterium. The bottom is for enrichment culture, which is about 60% of this Acetobacterium. And it seems to grow. And when I look at it, the removal of ammonium, on the left side, I have again the pure and enrichment culture. We remove ammonium. This is microelectrolysis cells. We added PFOA. There doesn't seem to be a negative impact in having PFOA in the ammonium removal. And when I look at the right side here, this is the amount of PFOA that I'm removing. This is my electrolysis cells where I have the active organism and ammonium. Here, when I have the dead cells or when I have no ammonium, I don't remove any of the PFOA. And this is a control where we have sort of our traditional incubation with just ferric iron instead of the microbial electrolysis cells. So it looks like we can use this bioelectrochemical reactors to degrade PFAS in the absence of ferric iron. I have a student that is going to defend very soon, but we sort of developed this further, and this needs more development. But we, with people from our material science group, they have developed some hydrogel anodes that seem to be working quite well, and looks like within a couple of days, at least the experiments we had so far, 3-4 days, we go from a part per million to no detect of PFOA. All right. This are the same. I see the same intermediates as we saw before. As the PFOA decreases, I see hepta, hexa, penta, and perfluorobutanoic acid appearing in these microbial electrolysis cells. Okay. Now let's go a step further and see how do we use this Acetobacterium in environmental settings. Can we detect A6 in PFAS impacted sites? So we went to Department of Energy sites where they used a lot of AFFF and see, is A6 present? Can we stimulate it? And if we can stimulate it, can we see some degradation of PFOA? And then the question is, how do we supply that iron to soils? Okay. So here, this is a subsample. This is a paper that showed up recently in Science of the Total Environment. What I'm showing here is where we take sediments from these AFFF contaminated sites, and we look specifically at sites that are acidic and iron rich because the organism needs acidic conditions. So we see A is when we have the sediments in DI water. B is where we add ammonium, and so F1 is where we add ammonium and iron. F2 is where we add ammonium, iron, and PFOS. Okay, so we see that when we add ammonium and iron, F1 and F2, we see that our Acetobacterium starts growing significantly. So we can stimulate it by adding ammonium and iron. And then when I look at the fluoride concentration, where we stimulate it, we start seeing a significant amount of fluoride production. And when I look at PFOA and PFAS concentration, we see that it actually over 60 days degrades maybe 80% of the PFOA and the PFOS. Okay. So it looks like we're able to stimulate in little slurry experiments by adding iron and ammonium, this A6 organism, and it does defluorinate these compounds. So next then is our iron. It's nanoscale ferrihydride or nanoscale goethite that we add. And what we learned is that when you coat, so the iron phases are positively charged, if we coat it with polyacrylic acids, and those are USDA approved food additives, so they're harmless. We change the charge of the ferrihydride from positive to negative, so that zeta potential becomes negative, and we can transport them through soil. So we've shown that when we have soil column experiments, we have a nice breakthrough. We can actually tune how much iron is retained by determining how much of the polyacrylic acids we coat to the ferrihydride. And then we did some column. What I'm showing then is Once we coat the ferrihydride with these polyacrylic acids, we checked multiple molecular weights. Control is the basic ferrihydride, and then from 2000 to 4500 molecular weight. We actually see there's a slight increase in ammonium removal once our iron is coated with ferrihydride. And the same thing happens with PFOA, the fluoride production. When I look at 40 days, the highest molecular weight polyacrylic acid has the most amount of PFOA removal and has the most amount of fluoride production. And we could show that what is actually happening is once you coat your ferrihydride with polyacrylic acid, the resistance of the electron transfer to the iron surface decreases by a good 30%. So it's actually helping the organism to transfer the electron and accelerates the whole process. Okay, so we did then, and I've been fine time. Column experiments were here to the left. I show soil columns from our AFFF contaminated sites, 2 different sites. And what I have here, and this slurry is my this is goethite, nanogoethite coated with the 450K polyacrylic acid. In the middle, we just feed to the columns the nutrient solution without iron and ammonium. And the right is just DI water simulating rain sort of hitting our surface. And what we've seen is, so what we ended up is we pumped all of these solutions into our column until we see breakthrough. And then we sort of incubated them. We stop pumping and let the column sit for 50 days. And so the organism starts growing and becomes active. And then we start to pump again and sort of retract the effluent. And what we see then is in terms of ammonium, where we added iron, and the red one where we added iron, we pumped iron into the column, we have the most removal of ammonium. When I look at fluoride coming out, both were added iron and ammonium. I have a significantly higher peak of fluoride than my DI water case, where I don't stimulate the organism. But it's a little inconsistent, or first you wonder, well, why do you see more fluoride coming out where you don't add the iron? And it's again because the iron complexes with the fluoride and we don't have a full recovery of the fluoride. So probably I'm producing a lot more fluoride where I had the iron, I'm not able to recover it. At the end of the experiment, we took the columns apart, we extracted the soil with methanol and analyzed for the PFOA. And what we see is that, changed colors here, but the blue one is now this amount of PFOA that we extract from the column that we added ammonium and iron. And we see that we had a significant decrease in concentration of PFOA compared to the column that was not stimulated, that we just pumped DI water through. Again, fluorine balance, not perfect. We get sort of for 80% recovery of the total amount of fluorine, right? So it shows now that if we have the organism if we have sediments where we believe the organism is present, even though if it's at very, very low numbers and rather inactive, that we are able to stimulate its growth by adding iron and ammonium, and that when we do that, it starts to actually defluorinate multiple PFAS. Okay, so let's revisit these dehalogenase genes in the A6 genome. We did experiments where we then looked at the expression of these dehalogenases as a function of the amount of fluoride that we produce. And what we've seen is that when we look at this reductive dehalogenase gene, the RDHA, there's a fairly good correlation between the amount of fluoride that we're producing and the expression of that gene. The fluoroacetate dehalogenase is a little bit, but it's more noise. And the haloacid dehalogenases, they're not expressed when we defluorinate. Interesting, when we add trichloropropionic acid, they are expressed, and we see a little bit of degradation of these trichloropropiononic acids. So they seem to be involved in dechlorination, not defluorination. We, with help from folks from our molecular biology department, we did some knockout experiments. So they knocked out the RDHA gene and the FCA gene. So the top line is the amount of fluoride produced in incubations of the wild strain. The middle one is where we knocked out the they knocked out the RDHA gene, and the bottom is where they knocked out the fluoroacetate dehydrogenase gene. And what we see is that mostly when we knock out the productive dehydrogenase gene, we don't have defluorination. Okay, there are a few exceptions where the experiment may not have worked. So what we conclude from here is that this reductive dehydrogenase plays a key role in the defluorination of PFAS. We just didn't know, is it one enzyme that does the defluorination, or maybe a series of enzymes that are needed? And we just managed to track one. So very quickly, I'm out of time. We know that this Acidobacterium is capable of degrading PFAS. It is present at some PFAS impacted sites, usually acidic. We have never seen it when the pH is neutral or higher. And it can be stimulated in anoxic laboratory incubations, where we then see the production of fluoride. And by overexpressing the RDHA gene in E. coli, we can see rapid defluorination. So it's interesting what typically takes us a month, 60 days, we see in 2 hours. And just final thought, I think we are now roughly where we were 40 years ago when we talked about chlorinated compounds, where there was debate, are they degradable? Is trichloroethylene degradable? Is it not? PFAS are probably more challenging than chlorinated compounds because the bond is much stronger, but we have much better molecular tools to sort of track what is happening. So this is lots of funders that got us where we are now. And why don't I stop here and answer questions? We'll take the time.